Hello friends, this is Seher from EasyPeasy, and the topic that we are going to discuss today is called as mitosis. So in mitosis, just like interphase, we have four subdivisions. That includes prophase, metaphase, anaphase, and telophase. Now let's talk about each phase in detail. So we will start with interphase, specifically the G2 phase, in which all the things are duplicated. So you can see there are two centrioles here, and we have a nuclear membrane. Nucleolus is here, and the chromatin material is spread inside the cell, and there is no visible chromosome. Once the cell enters the M phase, it will enter the prophase. In prophase, the centrioles are going to get apart like this. Nuclear membrane disappears, nucleolus disappears, and chromatin material condenses itself into visible chromosomes. If you look at this chromosome closely, you can see that the chromatids are joined tightly with each other with the ring type of structures here. These ring type of structures are called as SGO1 bound cohesion. And near the centromere, where we have the inner kinetochore and outer kinetochore, we have these red rings that is called as CENPU bound cohesion proteins that are going to bound these two chromatids tightly with each other. Once this prophase enters into the metaphase, so we will have a middle phase called as prometaphase. In prometaphase, all these SGO1 bound cohesion proteins that are these blue ring type of structures will go away from these chromatids, and the chromatids will look like this. So as you can see over here, all the blue rings are going away and the little is left in the middle. We can also see the green microtubules are getting out of the outer kinetochore tubule of these chromosomes. Once this prometaphase enters completely in the metaphase, we can see that the nuclear membrane completely disappears at that point, and the centrioles will make spindle fiber. And all these chromosomes that were scattered inside the cell will going to arrange themselves in the middle of the cell. At this point, all the SGO1 proteins will remove from the chromosome, leaving the both chromatids far apart. Now, this spindle fiber that is originating from centrioles are not that simple. We have different types of microtubules originating from the centrioles, making it a spindle fiber. So the first microtubule that we are going to talk about is the kinetochore microtubules. That is represented with the solid lines. These solid lines are going to attach themselves with the kinetochore tubules that are coming from the chromosomes. Then we can see the dotted structure microtubules, and they also have charge on it. And these microtubules will be called as polar microtubules. They're not attached with the chromosome, but they do have a charge on them. Then we have a third type of microtubules that are overlapping with each other like this, that is represented in red color, and they will be called as overlap microtubules. And last but not the least, there are some microtubules, as you can see on the side of the centrioles in green color. They will be called as astral microtubules. And as we discussed before, that the green microtubules are also originating from chromosomes, so they are overlapping like this. Over here in this picture, this green microtubule is coming from the chromosomes, and this brown microtubule is coming from the centriole. And they are overlapping just like in the movie of Avatar, where people were making bonds with their horses and dragons with their hair. Now let's discuss the functions of all these microtubules. Astral microtubules, as these are the tubules that appears initially, work as an anchor for the cell. When the spindle formation occurs inside the cell, It makes sure that all the chromosomes will align themselves in the center of the cell in metaphase. Then we will talk about the overlap microtubules and polar microtubules. Overlap microtubules are also called as interpolar microtubules. And during the phase of anaphase, these microtubules will elongate, providing the capacity for a cell so that the cell can divide easily by this process. On the other hand, the kinetochore microtubules that are attached to the centromere of each chromatid are going to shrink. So they will shrink and take that sister chromatid that is attached to each microtubule towards their pole. Okay, so these are the functions of microtubule. Let's discuss how they will apply their functions and get inside the anaphase. So in anaphase, firstly an enzyme will appear that is called as separase. This separase is going to cut the red ring type of structure that we discussed before, and these structures were CENPO bound cohesion proteins. They will degrade that cohesion proteins, and now the chromatids can go towards their pole easily. Next, as we discussed before, that polar microtubules are going to elongate and kinetochore microtubules are going to shrink. As a result, the anaphase will look like this. Now here, as you can see, all these sister chromatids are towards their pole. You can see that the polar microtubules are elongating and the kinetochore microtubules are shrinking. Now, the next phase is the last phase of mitosis, and that is called as telophase. In the telophase, the first thing that's going to happen is the reappearance of nuclear membrane. And the chromatids are going to disperse themselves again in the form of a thread like this. And nucleolus will also going to reappear. So now the cell division is almost done, and now it's going to enter the cytokinesis phase. In the cytokinesis, as we discussed before, that the cytoplasm of these two cells are going to divide. Now, the division of the cytoplasm depends on the type of cell it is. So if it is an animal cell, then there will be a ring type of structure appears that will be called as contractile ring. This ring will initiate the division of cytoplasm of the two cells. And if this cell is a plant cell, then there will be visible plates that will appear in the middle of these two cells, and they will be called as cell plate. These plates will be resulting in the division of cytoplasm into two cells. And then in the end, we have two daughter cells after mitosis. That's it for now. Thank you very much for watching this video. If you like it, please subscribe this channel.